This is IAQ Radio, Indoor Air Quality Radio, the voice of the indoor air quality industry, with your hosts, Radio Joe Hughes and the Z-Man, Cliff Zlotnick. And now, Radio Joe Hughes.
Good day and welcome to IAQ Radio Plus. It's episode 756. This week we welcome Carl Zimmer, the author of Air-Borne, The Hidden History of the Life We Breathe. Before we get started, let's thank our sponsors. They're the reason we can continue doing the show. IAQ Radio Association sponsors are IICRC, the Institute for Inspection, Cleaning, and Restoration Certification at iicrc.org. The Restoration Industry Association, RIA, at restorationindustry.org. The Environmental Information Association, EIA, at eia-usa.org. IAQ Radio industry sponsors are Particles Plus at particlesplus.com. Tramex Meters at tramexmeters.com. EMSL Analytical Inc. at emsl.com.
And now you can win a cool prize. It's time for the IAQ Radio Trivia question. Be the first to correctly answer. Simply email your answer to czlotnick@cs.com. Or if listening live, just text your answer from your computer. And now, here's the Z-Man.
Hello, everyone. Congratulations go out to John Lapotaire, Indoor Quality Solutions, Sanford, Florida, who was first to identify the country of Norway, from which the founder of the train company emigrated from. Here's today's IAQ Radio Trivia question. How many gallons of air does the average adult draw in and exhale every day? Back to you, Joe.
Thank you, Cliff. Carl Zimmer is the author of 15 books about science. His latest book is Air-Borne, The Hidden History of the Life We Breathe. Zimmer writes the Origins column for the New York Times. He's also a familiar voice on radio programs such as Radiolab and a professor adjunct at Yale University. Carl, welcome to the show.
Thanks for having me.
Great to have you here. Let's start with, what led you to be interested in this topic and to write the book Air-Borne?
I'd say that the seed was planted during the pandemic. I was working with a number of colleagues at the New York Times on covering this pandemic and writing about scientists trying to make sense of this new virus. How quickly does it spread? How does it spread? All these sorts of questions. And pretty quickly, there was a big debate about exactly how it spreads. A lot of us were, wiping down our groceries because the idea was that it was being spread through contaminated surfaces, maybe large droplets that people would like cough and, just hit things in the environment. But then there were other people who were saying it was airborne in the sense that it was spreading like smoke. And I was fascinated by how this debate played out and how eventually, the consensus emerged that COVID is airborne, and, the fact that it took a while. It just seemed like why wouldn't this be something that would get wrapped up really quickly? And so scientists said, well, you need to understand the history of this field, something I didn't really appreciate. And that kind of led me down into this whole world of science, not just about COVID, but about all the life in the air, what scientists call aerobiology. And, our atmosphere is full of life from, in these rooms where we're speaking all the way up to the stratosphere. And it's what scientists call the aerobiome. So it's just been a fascinating few years of exploring that world and trying to make sense of how we can protect ourselves and stay healthy in this world where we are inhaling living things all the time.
I love history, and I found it fascinating that some of the really early, when you think about some of the things they were doing before there was electricity, these guys were out there studying the aerobiome, and essentially before there was electricity. Can you talk to our audience a little bit about what you discovered in the early days of the aerobiome?
Yeah, there are definitely a few key pioneers. that I highlight. Some of them are familiar and some of them are not so familiar to people. But yeah, I mean, well before the age of computers and DNA sequencing and even electricity, they were trying to understand what's floating around us. Um, you had, microbiologists who were trying to understand the Great Irish Famine when suddenly all these potatoes started rotting, and they were saying, well, we think that there are microbes that are responsible for killing these potatoes. And other people said, no, 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 it's just bad air. And they said, no, no, these are living things, and we think that they are traveling through on the wind to get from one potato farm to another, and to travel from country to country. So that was in the 1840s. Louis Pasteur became convinced in the 1850s that there were germs floating in the air. And again, people just could not believe it. They would just say in print, like, this is just too fantastic. And so, in order to catch them, he created these flasks with very long, straight necks, and he would put sterile broth in them, and he would take them to different places. And he reasoned that the bacteria would fall down those necks and then be able to grow in this sterile broth. And he went into French streets in Paris, and he was able to catch germs there in the farmland in France, did it there, and even climbed a glacier. And even there, in that kind of rugged environment, he was able to capture some germs from the air. So, people would go to amazing lengths to try to understand this invisible world that surrounds us.
Can you give our audience a little kind of like a summary of the book in general? So, like a little overview, if you would. I'm sure a lot haven't read it. By the way, I've got a copy up here for our audience to check it out. Air-Borne, excellent read. 400 pages, which is not, that was a lot of work, Carl.
It was, yeah, I mean, trust me, actually, that manuscript was a lot longer than the final book. It's a really rich subject, but you definitely do need to trim it down. I would say that, just sort of the overview of the book, it is, looking at the aerobiome, this living world around us today, in a world where we have, a lot of people have become aware that, diseases can be airborne, COVID in particular, and then stepping back and trying to look at this history, not just of the early pioneers, people like Louis Pasteur, but people more recently, figures like William and Mildred Wells, for example, in the 1930s and 40s. But this history has dark sides to it too. Biological warfare was pretty much you taking aerobiology and insights into aerobiology and trying to make weapons out of that science. And I think that really diverted a lot of aerobiology research that has kind of harmed the science in the long term. But in the end, I hope that readers will, first of all, find it as remarkable as I do that, these ideas about life in the air actually have taken a really long time to settle in, as evidenced by the fact that, the World Health Organization flat out said in March 2020, COVID is not airborne. That kind of resistance has deep, deep roots. But, I also, in the book, hope that people will also, look up at the clouds and look at them in a new way because, the air is alive. The clouds are full of living things. There are bacteria eating clouds, and the bacteria might be actually making rain in the clouds. So it's a combination of that historical look at, the science that affects human health and also a new way of looking at the world, I hope.
You talked about, I think they call it like miasma. I don't know if I'm pronouncing that right or not. Can
Yeah.
you tell our audience a little bit about the early thoughts on miasma and how that changed over time?
So, about 2,500 years ago, ancient, Western physicians would think about health very much in terms of the air, but they believed that a lot of diseases, like the plague or malaria or influenza or other things, that they were all caused by what they would call miasmas, which it's kind of hard to really give a precise definition of that in modern terms because it's just our minds don't really slot into the ancient world. But you can think of it like corruptions of the air, that somehow the air would become disrupted, maybe by heat or the position of the stars or some other factors, and and then that air would drift along, that corrupted air, and if you breathe that in, you would become sick. It would disturb the balance of the humors that they believed was so important for health. So miasmas, uh, it was believed there were different kinds of miasmas for different kinds of diseases. Different species got different miasmas. It was this whole system, which is, very hard to make sense of because it's so alien to what we think of as in terms of disease. But it took hold, and in one form or another, it endured. In the Renaissance, when European scholars are rediscovering the Greeks and so on, they are very much embracing these ideas of miasmas. And even in the 1800s, the most prominent doctors and scientists are saying that, diseases like cholera are caused by bad air, by miasmas. And this really did not switch until the end of the 1800s.
You know, something happened along the way. Aerobiology was a pretty important topic and a hot topic for some of the early scientists, and then it kind of lost its way, I guess, and wasn't as important. What happened to kind of sidetrack aerobiology?
Yeah, to me it feels like it's kind of a cursed science. Like things just keep going wrong. And so, so people like Pasteur are demonstrating that there is life in the air. And, Pasteur himself said, he suspected that some diseases could be spread through the air, but, he didn't have any way of really nailing that down. And so, in the late 1800s, um, you know, medical researchers are starting to figure out really what the causes of a lot of different diseases are. And so cholera, for example, is not spread through the air. It's spread by bacteria, and those bacteria are spread in water. It's a waterborne disease. And so there turned out to be a lot of waterborne diseases caused by bacteria and other things. Um, Yellow fever and malaria, those are diseases that are spread by animals, by mosquitoes in particular. So you have vector-borne diseases and waterborne diseases, food-borne diseases, sexually transmitted diseases. So all these things that were used to be believed to be caused by miasmas in the air, they're getting pulled out of the air. And so by the early 1900s, there's basically, it seems to a lot of public health experts, well, there's nothing left in the air. We don't have to worry about the air. Relax, And they would actually write this, put this in writing, like, for thousands of years, we've been worried about infections in the air, and you don't have to worry about that anymore. What you do have to worry about is, washing your hands and making sure food is inspected and safe and things like that, which is all true. But the pendulum just swung so hard the other way that the possibility that, airborne diseases were real and were important and needed to be fought in the air got totally just buried.
and and that along with that being buried in the medical community, it was buried in universities. Very few people were actually studying it any further.
Oh yeah, yeah. No, I mean, pretty much no one was studying it. And it was just like every now and then someone would pop up and say like, like, let me check this out. While the medical community is shifting away from this, you also have other people who are studying life in the air from a very different perspective. You have people who study crop diseases. So people knew that, crop diseases could spread through the air, not just potato blight, but, there are plenty of fungi that attack wheat, like stem rust. And so a few scientists, primarily someone named Fred Meyer, kept saying like we need to really like map the life in the air. We need to understand like how far are these plant pathogens traveling? We don't know. We just know that they just seem to drop out of nowhere. And he did incredible work in the 1930s, showing that life could exist in the stratosphere, showing that it could cross oceans. Unbelievable work. But he did it all as a hobby because the USDA didn't consider it important enough to actually pay him to do. And so, yeah, it gives you a sense of just how it just aerobiology could never quite get the respect that it deserved, honestly.
Yeah, I found it interesting that they would put, balloons up and go in airplanes and open these capsules, essentially these tubes, and suck in some air, let some air suck in there, and then take it back down and take that air and put it on something that might allow it to grow and study what was in the air. And they found that there was life everywhere. Essentially, you couldn't go anywhere without finding life.
Yeah, and this really this work really blew people's minds because it expanded our sense of where life could be, what it was capable of. And so this was headline news. The fact that that Fred Meyer was, finding life, many miles in the sky, and not just in the clouds, but in the stratosphere. He got Charles Lindbergh on board. Charles and Ann Lindbergh helped him by, actually looking for life on their trip across the Atlantic. Amelia Earhart actually joined on as well. Unfortunately, her research was lost when she disappeared at sea. So yeah, it was amazing stuff that he was doing. But unfortunately, Fred Meyer died in 1938, and he was basically like creating the whole field of aerobiology. He even gave it its name in 1937, and then he disappeared, and with him, a lot of the momentum disappeared too.
Fred Meyer was one of the people whose shoulders we stand upon here. Another one was the Wells. I got their name here somewhere, but I'm sure you'll remember the Wells. But the most of our audience is somewhat familiar with the Wells-Riley model,
Mhm.
um, but not as familiar with the people who developed that. A lot of the story focuses on the Wells, and I'm wondering why they're such a big part of Airborne. I mean, it seems like they run through the whole book.
Yeah, no, they are incredibly important figures. And honestly, like when I mentioned, during the pandemic, I was trying to understand the context for the big fight over how COVID spreads. And some people said to me specifically, like, well, you should get to know William and Mildred Wells. And I had never heard of them. So, I was like, okay, well, I can always stand to get educated. So let me learn about them. And it wasn't easy because there's no biographies written of them. There's not even like academic papers published about them. They published a few things. William Wells wrote one book and they kind of disappeared after they died. William Wells died in 1963. So yeah, so I sort of went back and tried to make sense of who they were in their own time and their importance to later on. And like they really nailed a lot of the basics about airborne disease in the 1930s. They got a lot of it sorted out. They kind of identified the big research questions and they even went so far as to say, in the 1930s, they were saying, um, we have now figured out how to make water safe and how to make food safe, through the germ theory of disease and public health measures. We need to do the same with air. They set that as an important priority, and they even had an idea for how to do it, which is because they discovered that ultraviolet light can disinfect air. So, in addition to other measures like ventilation and so on, like they thought ultraviolet light, installing lamps would actually help. And, they demonstrated, quite effectively in a measles outbreak in 1940, where they had these lamps installed in schools, that, yeah, indeed, they can actually protect people from airborne infection. So, yeah, if they hadn't been forgotten largely, I think that, certainly the COVID pandemic would have played out very differently.
and it's a shame because they both died before their research became the foundation for a lot of what we do today. And that was sad to see. Let's if you could maybe describe what I thought was, and maybe I'm wrong, but kind of their most important experiment in showing that I think it was tuberculosis could be transferred via air. Could you kind of describe that for our audience?
Yeah, so this yeah, this might have been their most ex important experiment. I mean, they did a bunch of really significant research, and in hindsight, we look at these things and we say, "Wow, like they figured all this out." But the tuberculosis experiment actually was kind of an act of desperation. Um, and by 1948, things are really going badly for the Welleses. William and Mildred, they had worked for a while at Harvard. They just couldn't get along with anyone there. They got fired from there. They landed on their feet at the University of Pennsylvania, but after a while, the people there couldn't deal with them anymore either. And so they were let go. And the two of them had worked very closely as a married couple, but also as scientific partners. But they had a lot of problems, and essentially the marriage fell apart. So William Wells is thinking like, what can I do to get people to really embrace this belief I have that airborne diseases are important, they're real. And he decided that he would try to demonstrate that tuberculosis is airborne. He had done it with animals and animal experiments, but he wanted to show it. He wanted to show that people with tuberculosis, just by breathing in and out, could release the bacteria into the air, and then it could then spread like smoke. And it took about a decade, literally about 10 years, for him and a few of his remaining allies to finally get an experiment set up in Baltimore at a VA hospital called Loch Raven. And there they set up a ward with TB patients just lying there. They sealed everything off so that that the air wouldn't be leaking in and out. And there was just a ventilation system that would carry the air, including what the patients were exhaling, into a ventilation system and up into a facility on the next floor where they had guinea pigs. And guinea pigs are good models for tuberculosis. They get tuberculosis like we do. And so they would have the guinea pigs breathing in this air. And in other cases, they had the guinea pigs breathing in air that had been treated with ultraviolet light. And the guinea pigs would develop tuberculosis, you know, which is what William Wells had predicted. But not only that, but William Wells had predicted how quickly they would develop it, how often you would find a new guinea pig that had a little nodule in its lung. And he even got that right. The tragedy is that by the time these results were coming in, Wells developed cancer and also basically went psychotic and was paralyzed from the waist down. He had to be hospitalized, and the place he was hospitalized was Loch Raven. So he was literally like bedridden for a couple years while this experiment is slowly playing out over his head. And that really did finally get people to accept that at least tuberculosis is airborne. Unfortunately, the flip side of that is they said, yeah, okay, tuberculosis is airborne, but pretty much nothing else is. So that wasn't what Wells wanted. Wells wanted to say, like, okay, let me find one disease that I can really demonstrate this to people. And then people can think about other diseases like influenza or smallpox or all these other potentially airborne diseases. So it's an amazing story.
I can see how the medical community and other scientists prior to that experiment, were skeptical. But once that experiment was, completed and they showed that tuberculosis could be transmitted airborne, what was the reasoning behind thinking that was just kind of like a fluke? It was the only one. I mean, what led to this many years of people just denying that airborne infection occurred?
It's a really good question, and it's a really hard question to address because it's the sort of thing that people don't like write down. They very rarely like put into words like why they dismiss an idea. It's almost like, well, it doesn't even warrant even saying anything about. You just go on your way and you ignore something. Um, I think there was this incredible, there was this sort of, generation upon generation assumption that, diseases just spread in these other ways. And so it was almost like, there's no need, quote unquote, to explain diseases through airborne transmission. I mean, but the problem was that, that people were relying on sort of indirect evidence, and they were, in some cases, definitely getting mistaken. So it took these sort of agonizing experiments, agonizing in the sense they were long and took a lot of work to make them really rigorous, just disease by disease to demonstrate that, this disease is airborne or that disease is airborne. And with tuberculosis, tuberculosis is an unusual disease in the sense that like the bacteria have to get all the way to the alveoli, like the very tips, of the air, those passages in your lungs. Now that's very small there. Those passageways are very small. And so, William Wells realized like, okay, and the only droplets that are going to get that far probably have to be less than 5 microns just to fit in there. This sort of got taken up in the public health community as like, oh, so that means that the only droplets that can be airborne are less than 5 microns. So airborne disease is only spread by droplets less than 5 microns. Like that's complete. You would fail, an atmospheric physics class exam if you said that. Like, no, like the fact is that much larger droplets and particles can float around. And yet that became actually like incorporated into public health guidelines. You can see them, Centers for Disease Control would just say this. They kept saying it actually into the COVID pandemic. I mean, like, in other words, decades of misunderstanding Wells's work. So that's another reason for why people didn't really follow the Wells's arguments.
Let me ask, before we go to our halftime break, what did you find to be the most interesting kind of new thing that you learned during your researching and writing of the book?
I guess I was fascinated by how much life is going into the air all the time and from everywhere. So it is, in the morning, evaporation will pull bacteria, viruses, fungi, and so on out of the ground, and off of leaves and bark. Uh, in the ocean, every time you see a wave crash, it's actually releasing lots of tiny droplets, sort of in the way I'm releasing droplets by speaking. Waves release their own tiny droplets, some of which have life in them, and they go up into the sky. They can go up into the clouds. So, yeah, I think like starting to appreciate like, oh my goodness, like this planet is constantly essentially breathing and releasing life into the air is it definitely changes the way you think about the world.
It definitely does. Let's get into a little more on that after halftime. We're going to stop and thank our sponsors. We'll be back with the second half of our interview. We've got Carl Zimmer, author of Air-Borne, The Hidden History of the Life We Breathe. Our association sponsors are the Environmental Information Association. EIA's multidisciplinary membership collects, generates, and disseminates information concerning environmental and occupational health hazards in the built environment at eia-usa.org. The IICRC, a nonprofit standards development and certifying body for the cleaning and restoration industry, iicrc.org. The Restoration Industry Association, the oldest and largest nonprofit professional trade association dedicated to providing leadership and promoting best practices through advocacy, standards, and professional qualifications for the restoration industry at restorationindustry.org. Industry sponsors are Particles Plus, feature-rich particle counters and air quality instrumentation. Count on us, particlesplus.com. Tramex Meters, innovation in moisture detection and humidity monitoring systems since 1974 at tramexmeters.com. EMSL Analytical Inc., providing high-quality, full-service laboratory services since 1981. EMSL.com. All right, we're back with Carl Zimmer on his book Air-Borne, The Hidden History of the Life We Breathe. Carl, I got a note here that, and I noticed you mentioned in an email, we need to find a balance between protecting ourselves from dangerous pathogens and allowing the aerobiome in. I think this is something that, our audience has a lot of people in it that help people with indoor environments and after disasters or when they have water damage related issues or ventilation or whatever the case may be. And I think finding that balance is a little tough for them at times as well. Can you elaborate on that statement?
Sure, and I can understand, the where folks are coming from. I mean, if your house is, full of a highly virulent pathogen, if you've got, black mold floating around and you want to get that out. You want to figure out strategies to clear it out of the house or to clear the air, and you don't want to mess with that. Um, but the fact is that every day from birth we breathe in living things. We just do. I mean, you can't not breathe in living things because the air is full of them, like I was saying before, and it's not just pollen during hay fever season. It's just every day the earth is sending up vast amounts of microbes. And, like there is some research, and it's again, quite preliminary, but like researchers are trying to understand, well, what is the effect of our bringing in the aerobiome into our bodies every day from birth? What happens? Why is it that our immune system doesn't like attack at all? And, there's sort of a broader study of our interactions with microbes, the human microbiome more broadly, which is giving us, some really important clues about our relationship with other species. And so, when, it's great that we have antibiotics. If you have a severe infection that's going to kill you, like if you can take a pill that will just knock out that thing that is about to kill you, that's great. Unfortunately, antibiotics, at least most of the antibiotics that you can get these days, they're really indiscriminate. Like they kill lots of microbes, and so they will actually disrupt the microbiome. And so when you have kids, for example, who get lots of ear infections and they take lots of antibiotics just over and over and over again, they really keep killing off a lot of the regular inhabitants of their bodies. And that can lead to a disturbance in that ecosystem, and that has been associated, I should say, with a lot of disturbances of the immune system, things like allergies, for example. And there's a thinking that our immune system has evolved to basically mature by having a constant interaction and interplay with microbes, so that it essentially, quote unquote, learns how to keep us healthy while tolerating a certain amount of visits from the outside and visitors who just sort of set up shop inside our bodies. The air biome, something like that might be similar. So it might actually be good for us to be breathing in things. Some people have even suggested like, the air in certain places, like, a healthy woodland. That's a good place to be breathing in the kinds of microbes that are floating around in the air. So again, this research is in its very early stages. So I'm not telling anybody, I'm not going to be giving anybody any medical advice about this. But it does address this fundamental fact, almost like a paradox that, what we breathe in, sometimes it can kill us, but most of the time it doesn't.
It's an interesting conundrum, I guess, because we had Dr. Jeff Siegel on the show. He's up at, I think, University of Toronto,
Mhm.
and at the end of the show, we asked him, what could you do to help make your indoor environment healthier for your kids or something along those lines? And he basically said, get a dog, which is
Mm.
kind of counterintuitive. You think,
Yep.
well, I'm allergic to dogs, so it's probably not the best thing for me. But
Right.
so get a dog. Make sure your kids are exposed to a lot of different microbes and different exposures when they're young, so
Mhm.
that they can fend them off when they get older, I guess.
Yeah. Get a dog, play outside, roll around, you know.
Get
Uh,
some dirt in your hands. You
yeah.
know,
I mean, now obviously, like, I mean, you you want your kids to wash their hands before they eat. Like, we're not talking about being filthy, but like you also don't want to put children in a bubble away from these other species that we live with at the very time where they are developing their immune systems. So when we're adults, if we have allergies or other immune system issues, we may, that time for like getting a dog, to help you, that time might be past because now your immune system is mature. And then you might need to use other strategies. But yeah, certainly you have to remember for children in particular, it's a crucial developmental period. And it's a developmental period that seems to like involve not just our own species, but our interaction with other species. And some of those species are like living on dog hair, or farm animals, like kids who grew up on farm animal and on farms, have much lower rates of things like allergies and other immune disorders. It's just that's the fact.
asthma.
And so some of it is probably from direct contact, getting things on your hands and kind of wiping them around on you. But a lot of it might be just inhaling, you know, and either those bacteria or fungi or even viruses might kind of establish themselves in the airway, or they might, and we might sort of like cough them up and then swallow them, and then they become part of our gut microbiome.
Now, you talk to a lot of interesting people in this book, and I don't know if there's some we've had on the show. Dr. Shelley Miller, I noticed, was in the book. Rich Coursey, we've had on many times. I'm wondering, were there any people you wanted to talk to, but they didn't agree to talk to you for this?
I generally had pretty good luck, I would have to say, in just reaching out to people, which was nice. And even some people who would say, you know, I, you know, I was taught not to take airborne disease seriously, and I was wrong, and it's good to learn. Uh, People who are willing to acknowledge those things. There were people, I would say that, it's a general issue, not just working on a book, but in reporting. It can be difficult to speak one-on-one with people at public health agencies, World Health Organization, Center for Disease Control, partly because, they have a lot of just standard, rules like, you don't just call them up, you have to call a press office, then they want to know what you want to talk about, then they just decide whether they can even respond and so on. That's always a challenge. There's no question about that. So, sometimes then in those sorts of cases, you have to rely on sort of public record stuff, press conferences, videos on YouTube, web pages, various documents and so on. And, there's lots of stuff out there that you can rely on then.
Who was the most interesting person you talked to?
Hmm. The most interesting person I talked to.
And helpful, I guess, too, interesting and helpful.
Oh gosh. Well, I mean, I would say like Donald Milton of the University of Maryland. I don't know if you've had him on, but
I'm trying, Carl, help me.
okay, well, I mean, I became aware of Donald Milton. I think he was the first, aerobiologist, if I can call him that, that I became aware of. And this was way before the pandemic, because I came across a study he had done on influenza. And what really just caught my eye was this device he had created. He called it Gesundheit 2. And basically, you get people who are sick with influenza tested positive, and they sit in a chair and they sort of stick their head in a giant steel cone. And, it's the weirdest looking picture of, like, on the side, like, because you don't see their face. It's just like suddenly, like, imagine your face had a giant steel cone on the front of it. And then he would, I wanted to know what he was working on. He's like, well, I'm just having people breathe, and then I'm actually capturing the droplets, and then I'm seeing what's in the droplets and so on. I was like, oh, that's very interesting. Uh, and so when, early in the COVID pandemic, I reached out to him and I'm like, so what do you think about COVID? Because he had been studying influenza, and he was like, I don't like it. He didn't like the early signs that he was seeing. He was, when he would look at X-rays that were taken of children who got COVID in Wuhan in China, in January 2020, like early cases. And he said, like, this looks like something where, the virus has gotten deep into the lungs, and if it's getting deep in the lungs, that tells me that it's in lots of little droplets. That tells me that this could be airborne. And then we're going to have to deal with this differently. And yeah, and then Milton introduced me to the Wellses and so on. So I owe a lot. I've learned a lot from my conversations with Milton.
Now, um, I think a lot of people forget that COVID was not the first coronavirus that we dealt with. And the first one came in, I believe it was the early 2000s, in that era, somewhere around there, maybe 2010. And it kind of came and then it petered out. What's the theory behind why that one didn't take hold like the 2019, 2020 version?
So coronaviruses have been known about at least since the 1960s. That's when scientists actually identified coronaviruses under a microscope and saw that they all had this very distinctive shape. But at the time, it seemed like all they did was they caught, in humans at least, that they just caused colds. So, then by the 1990s, a lot of infectious disease experts were warning about, the next disease, new diseases, emerging infections. And they had a list of things that they were worried about. Different groups of viruses and other pathogens that could emerge and be the next big killer. Coronaviruses were not on the list. So then in late 2002, people in certain parts of China start getting sick with pneumonia, which doesn't really seem to fit pneumonias that people are familiar with before. It's not influenza. It's not a bacterial infection. And it turns out this is a coronavirus that causes this potentially fatal pneumonia. And that came to be known as SARS. And that was, it was really terrifying because this thing was spreading out of control, getting into Hong Kong, and then from Hong Kong to a bunch of other countries, including Canada. And at the time, I mean, a lot of the world was terrified of this thing. And, because here was this emerging disease that people had warned about, and it's a disease new to science. Now, we were lucky in the sense that, quote unquote, only, I think around 800 people died of it. The reason that more people didn't die was because it turned out to be pretty manageable once you kind of understood the course of the disease and could recognize symptoms and you could isolate people. Um, along the way, there was, people were just sort of assuming that it kind of spread through contact and short distances and so on. But a few scientists actually noticed that, no, this looks like it might be airborne. And they were people who rediscovered the Wellses, and they kind of, would then continue to do their work in the years that followed. Fast forward to I think it's like 2012, I believe, another coronavirus spills over. This one is called MERS, Middle Eastern Respiratory Syndrome. This is um, pops up in the Middle East in places like Saudi Arabia, and people are getting it from camels, and then they are sometimes passing it on to other people. MERS also turned out to not be, you know, a terrible, terrible disease because it really only could thrive in hospitals, infecting people with pretty weak immune systems. So you still get cases of MERS now, but I mean, we're just talking in the hundreds. Then we get the third coronavirus spilling over in a significant way, and this is SARS-CoV-2. And they were just it's related to the virus that causes SARS, but it's different in some ways. For example, people can be very infectious before, days before they themselves feel symptoms. And in many cases, they never feel symptoms. So you can have like lots of people going around and being fine and looking fine and getting lots of other people sick. And because it's airborne, it just those people go into the right spaces, restaurants, churches, what have you, and boom, they can just that's how this pandemic took off.
In the book, there's a chapter on disease X. Are we ready for the next disease X, Carl?
Uh, well, I don't think we're as ready as we should be, and I mean, and I think there are, and that's not because we don't. It's not necessarily because we don't have all the technology we need, but because there are just decisions that political leaders have made that are not going to help us. So the United States is not part of the World Health Organization. So that is going to make it harder for the world as a whole to be monitoring for the next disease that spills over from animals. That will happen, and that kind of fragmentation is going to make it harder. We we need to have, a whole system of, there was a big program that the United States had going for developing antiviral drugs for the disease X, for the next pandemic that we don't even know about. And the basis of that was let's design drugs that can work against whole families of viruses. So you don't need to know the details of one species. So you could have imagine a drug that worked against all coronaviruses, worked against SARS, COVID, MERS, and whatever comes next. That program was just killed in the last couple months. So, if we keep doing these things, we are going to be less ready for disease X.
One of the things that going back to the Wells discussion on their work, one of the things that you mentioned and that I wanted to make sure we bring up again is UV lights.
Mhm.
And you seem to be pretty impressed with the potential for UV lights. Now, there's also some like with anything else, there's pros and cons. UV lights break things down. They may produce some indoor environmental issues as well as solving others. But I'm wondering, um, most of the people you talk to seem to be pretty, think maybe that we weren't using that technology enough. Is that accurate to say?
What I find really interesting about ultraviolet light in particular is that, there, while there are people right now who are working on, developing and testing ultraviolet lights as a possible way to disinfect air, um, this isn't new at all. It's been about 90 years since William Wells and his colleagues demonstrated that you could kill influenza viruses and tuberculosis bacteria and so on in the air, in droplets floating through the air. And so, it's not, and so the fact that, a few people have continued to try to to carry on that work and to talk about using things like ultraviolet light in certain places is really striking to me. So in the 1980s, for example, I write about Ed Nardell. He's a tuberculosis doctor at Harvard Medical School, and he was in charge. He was in charge of trying to deal with a tuberculosis outbreak in homeless shelters in Boston, and he actually got in touch with one of William Wells's students, Richard Riley, who himself was rather old at this point. And, Riley explained to him about how ultraviolet light can potentially work. And they put ultraviolet lights in these homeless shelters. And, their evidence suggests that it worked pretty well. Um, so I find it fascinating that that thread continues even to today. Like, so Ed Nardell actually invited me to come hear him sing at a cabaret. He loves to sing and perform these songs, and he goes with some friends, and they have a great time. And he has these UV lights installed in this space, so it's safe from COVID. Like, and you don't need to wear a mask. It's okay. But certainly I'm not. That doesn't mean that I'm saying like UV light is the only way to deal with air. And certainly like when when these experts like Lindsay Marr and Joe Allen and other people have come forward with proposed mandates for indoor air quality, they're saying like, here's the number of exchanges, we want per hour, or, here's the CO2 level we think is right. And here are like a variety of ways that you could get there. And then, you've had plenty of guests who've talked about all the different ways that you can get there. So, I think that UV light could be a really useful option in places where other things don't work. Will scientists have to figure out about the risks caused by, this sort of essentially creating indoor air pollution? Sure. But I don't see that as like as a UV killer. That's a problem that presumably scientists are going to be able to overcome.
Yeah, it's like we talked about earlier, it's a balancing act. Would you rather breathe a little more of this byproduct of UV light or coronavirus? I
Mhm.
mean,
Yeah,
sometimes
and
those just,
and yeah, and there may be there may be different particular times when, that calculation shifts. So if we have, disease X showing up and it's airborne and rates are very high and there's not going to be any vaccine for months, then, you might say it would be nice if that was an option. Like, okay, I'm not going to be able to retrofit this, this school. But if I stick some UV lights here, they could be very effective, and I could use that as a strategy. But that it's going to be part of like a hopefully a more sophisticated way of dealing with the reality of airborne disease with these very sophisticated models that are being generated these days, sort of building on the old Wells Riley models. I think we can. There is a potential to deal with these things in a much more effective way rather than just cowering in fear.
and I think Kishore brings up a good point. The proper installation of UV lights is important to avoid harm to humans and for its proper efficacy. So it's not as easy as just putting some lights in. You got to do it in the right way. And now there's a
Yeah.
discussion about far UV versus regular.
Mhm.
So there's a lot more. Let's go to the roundup, Grayson. We're gonna wrap things up here. I want to give Cliff a chance to jump in. And as always, we want to thank Tramex Meters at tramexmeters.com for sponsoring the roundup. All right, Cliff, let me turn it over to you. I think you had a follow-up.
Yeah, Dr. Zimmer, in your investigations and so on and so forth, did you happen to run by the name of Alan Zelikoff at all?
That doesn't ring a bell. I mean,
Okay.
there obviously
No,
lots of people doing important work, and so I wasn't able to talk to everybody.
no problem. The reason I mention it is, so number one, he's a physician. Number two, he's really an expert in pathogens and bioterrorism and stuff like that, worked for the government and so on and so forth. The one thing that he said that struck me the most of all these different things that could be done. And Joe and I have had him on the show a number of times. Every time there was a suspected pathogen as being the big one, we would have him on and he would talk about the same thing over and over again. The one thing that he talked about is they teach all sorts of things in medical school except statistics. And he could tell from the infection numbers which one of these was the real one. Okay. And he knew that COVID was the real one and the other ones were not. Knew very, very early on just by looking at these infection numbers. And he just said if they only taught this in schools, it's known, people know about it. And I think it's much like Wells Riley or whatever. The information is out there. And, for some reason, people just have a tendency to ignore it. So I just wondered whether any of the people that you talked about, just talked about using statistics to tell early whether or not it was the real disease X.
Yeah, that actually happened, with COVID, with a number of people, would look at those early case reports and say this doesn't look great, and because the early estimates were that, one person might infect a number of other people under the right circumstances. And yes, so, that's what you don't want. And that's why measles is becoming such an issue again, because, it is airborne and incredibly contagious. And so, yeah, when you see a new disease and you see some cases, but you don't see, it's hard to see how one person is really infecting many other people, then that can, give you some hope that there are ways that you can contain it. And yeah, and there are people now who are like trying to actually combine those sorts of statistical epidemiological models with the sort of Wells-Riley type more physical models to actually like think about, get a fuller idea of like, what can happen inside of a building or a room, or, like how can we use all of our predictive powers to understand these new diseases as fast as possible.
And I just think I had one other comment really with the UV lights. And I think the one thing that we learned is that in schools and buildings, a lot of money was spent or set aside to improve the HVAC systems in all these buildings and improve the filtration and so on and so forth. And a lot of times that was either never done or not really effective. And what was effective was these personal units, Corsi Rosenthal boxes and so on and so forth, and putting them in schoolrooms. And I think it's the same thing with ultraviolet light. I think a lot of money has been looked at, installing it into the HVAC systems in buildings, but in hospitals, putting those UV lights in patient rooms, and dealing, focused at the ceiling, and have been proven effective for a long time. And I don't know that the larger, more expensive systems that are installed within the HVAC, have been proven to be effective, throughout an entire building. So,
Right. Well, yeah, and I mean, and those questions will benefit from some of these more complex models that are being designed. Like, how, what is the concentration of the pathogens that are moving through an HVAC system? Are you really like, killing that many viruses, for example, as opposed to having the UV lights like in a room right where people are exhaling, and so where and the concentration is denser. Maybe you're going to be able to do more good in the rooms as opposed to in the HVAC systems.
thank you.
Carl, do you have another 5 minutes here? Or you got to
Sure,
run.
sure. I'm good.
Okay, I've got several questions I'd really like to get to. One is about silos, essentially, that I think your book shows the importance of getting people out of their silos. Um, this is still a problem in the indoor air quality world. We've got, HVAC contractors, we've got restoration contractors, we've got people who do industrial hygiene type work, et cetera, and they all kind of look at things a little bit differently. And I'm wondering, why do you think the government resisted so hard in calling COVID airborne? Or were they just following the silo of the medical community?
I think that it was, a combination of things, and that's one of the themes in the book. It's like, how could we end up in a situation where, the World Health Organization says in March 2020, COVID is not airborne, and then almost 2 years later they say, yeah, it travels by long range transmission. And I think that, I think siloing is a big factor here because, in the early 2000s, you had a small kind of renaissance of, people who followed William and Mildred Wells. And, they were trying to make as much noise as they could about, the importance of thinking about these issues, and pointing out the flaws in the public health guidelines. Uh, and they weren't being listened to. And a lot of them were not in those fields of, infectious disease. They might be people who studied air pollution more as physicists, And so yeah, so you have these people in these silos, and they weren't really inclined to listen to each other. And that was a big problem.
How do we break that down? How do we combat that problem?
It is maybe it's part of human nature. It's certainly part of the way science works. Like people like spend years getting a degree in a particular program. They learn a particular language. They learn a particular way of doing things. They're not learning about what's happening, in, the next building over on campus, where there's a whole different culture being created. So you have to find ways to create interdisciplinary connections. Right now, again, to its credit, the World Health Organization is actually supporting just exactly one of those collaborations. I've been talking to people involved in it where they are, you literally have physicists from CERN, from the place where they smash atoms, who are using their deep knowledge of physics to create, beautiful, detailed models of buildings and rooms in which there are people who are inhaling and exhaling and tracking the concentration of all these things, and then they're working with epidemiologists and infectious disease experts to make those models realistic for diseases like COVID. And that's happening right now. That's great, but it has to keep going.
Now, please tell our audience about, I know this is one of the things that's very interesting to you. Some of the new research on indoor air quality, such as real-time gene sequencing, aerobiome detectors, such as the ones being supported by grants from the Advanced Research Projects Agency for Health. I had to look that one up, ARPA H, and their Breathe program.
Yeah, so this has to do with, how do you capture things out of the air? So, back in the 1800s, you had someone like Louis Pasteur just holding flasks over his head. Like that's not a very effective way of doing it. William Wells invented something called the air centrifuge, which is a very clever way to basically run a fan to pull in air into a glass cylinder and then capture things with centrifugal force. Um, Today, there's a whole new generation of, very sophisticated air samplers, which can not just, capture microbes, but um, then preserve them in such a way that they can be then analyzed for their DNA or their RNA to understand, to look at their genes to figure out what are these things. No, so those things are very experimental right now, but there is a project supported by ARPA-H, which is very forward-looking. But basically, what they want to do is create a device that you could use to detect pathogens, kind of like the way you have a carbon monoxide detector. You stick a carbon monoxide detector on your wall and you don't think much of it. It's pretty cool. It can detect one particular molecule and let you know while not bothering you about other ones. So imagine that there was a detector that would be constantly sampling the air and might say like, hey, we have a problem here. We have this particular strain of influenza in the air. You should know about this, or COVID, or hey, there's some virus here that we've never seen before, and this is concerning. And here's the genome sequence of it, things like that. That is work that's actually going on right now. I mean, It's probably going to be years before that will be stuck on the walls of hospitals or schools or homes. But that would be a real revolution in this effort started by people like Louis Pasteur.
I think Kishore brings up another good point. Mobile phones will be the future detectors of indoor air quality. I think that's, they detect everything else. Why not indoor air quality as well? Carl, before we go, I want to thank you for both the book and for joining us today. Is there anything we missed that you'd like to add or any final thoughts for our audience?
I think we covered a lot of ground. I really appreciate being able to talk to you about this, and I hope it's relevant to, your audience who are obviously dealing with indoor air quality every single day of their working lives. You know, I just, my hope is that, even for people who deal with these issues all the time, that they will enjoy this book and they will learn something from it. I mean, I certainly learned a lot.
That there was one other question. All right.
Mhm.
If you had to find someone to help you with your indoor air quality, let's say you had an issue in your building at Yale, where you teach or in your home, how would you go about finding that person?
Great question. I don't know. I don't know. I suppose, at a college, I suppose you could go and talk to the people, who are in charge of the physical plant. I will say I was surprised when I came into my classroom in the fall. I was in a new classroom space and had been renovated, and the students were sitting down, and I looked over at the thermostat, and I'm like, wait, like I see the temperature, but I also see the carbon dioxide concentration. This is awesome. This is telling me about how safe this room is. So that gave me a little bit of hope. So whoever put that carbon dioxide monitor in there, like I need to talk to that person.
Absolutely. Well, Carl, thanks again for joining us. We appreciate it. This is Radio Joe Hughes saying thanks to this week's guest, Carl Zimmer, author of Air-Borne, The Hidden History of the Life We Breathe. Of course, my co-host, the Z-Man, Cliff Zlotnick, Grayson Gone Fish and Fisher at the controls. Most importantly, our sponsors and our loyal audience. We'll be back next Friday at noon with the next episode of IAQ Radio Plus.
For IAQ Radio, I'm Spike Reel saying thanks for listening.